I first wanted to give everyone a brief overview of NIST because many may not be familiar with us. We are a non-regulatory agency in the United States Department of Commerce. We are tasked with developing standards that support international trade. And we've been around since 1901 when we were founded as the National Bureau of Standards. We are a relatively large organization, 3,000 employees, 1,800 of which are scientists. We have almost an equal number of associates and facilities users on our 2 campuses, one in Gaithersburg, Maryland, and one in Boulder, Colorado. We also have 5 joint institutes around the U.S. that have specific focus areas. NIST's mission is to promote U.S. innovation and industrial competitiveness by advancing measurement science, standards, and technology to enhance economic security that improves quality of life. We do that through numerous services, including over 2,200 scientific publications annually. We produce 1,100 reference materials and 100 data products, and we also perform 18,000 calibrations a year. Part of NIST, the Material Measurement Laboratory, is the U.S. National Reference Lab for Measurements in Chemical, Biological, and Material Sciences. In the MML, we make up about 1/6 of NIST's employee population, and we produce nearly all of the NIST reference materials. A significant portion of the NIST reference materials produced are used as tools for laboratories to ensure the quality of their chemical measurements. 2 categories of these reference materials are calibration solution reference materials and matrix-based reference materials. In the center of this slide is a diagram depicting a typical laboratory workflow, beginning from calibration through determination of a measurement value in a sample. NIST reference materials can be utilized in different portions of this workflow, including in calibration and in validation and verification of the overall analytical measurement process. Calibration reference materials typically are neat chemicals or solutions that can be used to determine response and calibrate the response of an analytical instrument. Validation and verification reference materials are natural matrix and carried through the entire analytical process in order to identify any biases throughout that entire procedure. In addition to reference materials, NIST also offers a number of quality assurance programs that produce materials that can be used also in validation and verification of methods. From the outside, a quality assurance program and a proficiency testing program have a lot of similarities. However, quality assurance programs have a very specific goal of improving laboratory comparability and exploring laboratory challenges. They are less about evaluating participant performance or meeting those accreditation requirements. There's much more of an educational focus with a quality assurance program. The outcome of a quality assurance program is insight into that method comparability and, in theory, improved laboratory practices. There is less emphasis on pass-fail scores, such as Z-scores, and potential corrective actions. Quality assurance programs are designed for new methods, new analytes, and new samples, or new staff and analysts, methods that are not yet part of the accreditation scheme or new exploratory analyses for a company. Proficiency testing is better for more established or routine tests or established staff and analysts. At first glance, a quality assurance program strongly resembles a proficiency testing program. Laboratories sign up for studies that are offered. Samples are prepared and distributed to those laboratories. They analyze the samples and submit their data to NIST. NIST then analyzes the data and provides feedback in the form of data tables, graphs, and written reports. As shown in this graph here, on the bottom axis, all the laboratory information is coded. That code is known only to the laboratory and to NIST. Therefore, no identifying information is provided in the general report. The data points on the chart here are color-coded, in this case related to the sample preparation approach used by the laboratory. We also color-code data points with respect to the analytical approach, such as LC-MS or GC-MS. This allows us to identify any methods that are providing a specific bias, high or low, or greater variability as a general trend. Laboratories can compare their performance to the performance of other laboratories by comparing their data point to other data points. You can also compare their results to the consensus of all other participants, as shown in the green shaded area and the dark blue solid line in the center, which is the consensus range and the consensus mean. In many cases, NIST also provides information about a target value determined based on either NIST results or previous information about the sample. This is a touch point for accuracy. We do offer upper and lower ranges of tolerance around the consensus, which are used to calculate Z-scores for those laboratories that do wish to use quality assurance program participation as part of an accreditation scheme. I also wanted to highlight a few of the lessons learned through quality assurance programs at NIST over the years. I mentioned at the beginning that the focus of these programs is education, both for NIST and for the participants. So here's an example from a couple of studies we ran in 2010 and 2012 through our dietary supplements quality assurance program. In the first study on the left, we asked laboratories to measure the total amount of catechins in some green tea samples. And when we plotted the results for one sample versus the results from another, the data points trended upward and to the right. And typically when we see this kind of a trend, it indicates that there is a potential calibration issue going on in the laboratories. Laboratories reporting low results are reporting low for both samples, and laboratories reporting high results are reporting high for both samples. We repeated this study again in December of 2012, and as you can see in this similar graph, the data points are now much more randomly distributed. We hope that this is because through the education of the laboratories in the report that we prepared, discussing this potential calibration issue, laboratories were able to evaluate their calibration materials and correct for any impurities or biases that they were observing. Here I have a couple of additional examples of lessons learned from our quality assurance program. On the left is an example from the micronutrient measurement quality assurance program, where you can see that over time, through the use of the quality assurance program and related reference materials that were produced through this program, the precision and accuracy are improved for laboratories participating. On the right-hand side is an example from our vitamin D quality assurance program, where we were able to identify method challenges for immunoassay approaches in red compared to chromatographic and MS-based approaches in black. As you can see, the overall mean for the immunoassay results is lower than that for the LC-MS results. And through education and feedback to the participants, laboratories are able to improve the methods that they use and understand better the data that they report. So with that, I just want to remind everyone that NIST offers a number of tools for laboratories to ensure the quality of their chemical measurements. Two of those major tools are the reference materials and the quality assurance programs that we offer. I've provided here my email address. If you have any follow-up questions, of course, I'll be on the Q&A after, and also a link to our website for purchasing reference materials and participating in our quality assurance program. So with that, I will turn it over to my colleague, Brent Wilson, who is going to talk with you a little bit more about the reference material and quality assurance program development going on at NIST directly related to cannabis measurements.
Thank you, Melissa, and thank you to the attendees for the webinar today as I look to demonstrate some of the tools we are developing at NIST for the cannabis community. Cannabis can be split into two separate categories based on its concentration of THC. Hemp, which is primarily based on cannabidiol, CBD, is federally legal when the concentration of THC is less than or equal to 0.3% on a dry weight basis. Marijuana is federally illegal when its total THC concentration is greater than 0.3% on a dry weight basis. The cannabis community is very diverse. As I demonstrate here in this puzzle diagram, where it includes regulators, including FDA, USDA, or state regulators, product developers, including farmers, formulators, and dispensaries, contract testing laboratories for hemp samples and or marijuana samples, and forensic laboratories, such as federal, state, and county and local police department crime labs. At NIST, we feel like we are a key component of the cannabis community as we have a direct relationship with all four sectors of this puzzle diagram, and that we can provide the necessary analytical tools to help the community. The two primary tools under development at NIST include a quality assurance program, CannaQAP, and cannabis reference materials, with a focus on oil samples, plant, including hemp and marijuana, extracts, concentrates, /distillates, and edibles. The first exercise of CannaQAP will include 2 different hemp oil samples. These were selected because they would require minimal sample preparation by the participants, and the material is homogeneous to start with, which is a major issue when dealing with plant material. This will allow us to do a true evaluation of the analytical method itself, which could include LC-UV, LC tandem mass spec, and GCMS. The measurements that we're targeting in this exercise are total THC, which is calculated based on your delta 9 THC and your THCA values, which the conversion shown here on the right. And also CBD and 14 other cannabinoids that are reference names are commercially available for. Now, it is important to note that the participants aren't required to report all values for all of these analytes. If your method only focuses on measuring THCA and delta 9 THC, then we recommend you just submit data for those 2 measurements. Here's the current schedule for exercise 1 of CannaQAP. At this point, we're in stage 3, where we're asking participants to create accounts so that they will be automatically notified when they can sign up for exercise 1 later in June. We are also encouraging people to go ahead and create accounts even if they don't intend to take part in exercise 1. This will also allow them to be notified automatically of future exercises. In July, NIST is hoping to send the samples out to all the participants, and will make additional measurements at NIST to characterize the samples. Participants will be allowed 2 months to make sample measurements and then enter in their data into the CannaQAP website. Included in this will involve what type of analytical methods they use, sample preparation, and any type of instrumental specifics associated with their method, to allow NIST to essentially evaluate potential bias from one method to another. Afterwards, NIST will release preliminary data tables of all the data submitted to the participants so that they can confirm and make sure that their data is reported correctly. Then in October, NIST will provide a full detailed report that will summarize the data and compare different analytical methods and provide conclusions of the overall program exercise. As part of developing CannaQAP samples and cannabis reference materials, NIST will be developing a third tool for the cannabis community, analytical methods. These methods will include sample preparation, LC-UV, LC tandem mass spec, and GCMS. I'm showing here a general diagram of the 2 main matrices we're focusing on first. in this case, a hemp matrix plant material and a hemp oil sample. The focus is to measure cannabinoids in these two matrices. And here I'm just showing where you can look at the different chromatographic response, where here on the top you can see a sample that's most likely going to be in this case, a marijuana sample because you have a higher delta-9 THC concentration. In the bottom, you have a much higher CBD concentration, representing a hemp sample. As part of Exercise 1, NIST will provide analytical measurements for the two hemp oils. In this case, I'm showing you an example graph of what that data could potentially look like, including the 3 different analytical methods NIST will use. We will provide mean values for each of the analytical methods and a plus or minus standard deviation associated with their measurements. Each method will include measurements of 3 aliquots and 3 replicas of each aliquot. So in total, we will have a mean value based off of 27 measurements. And we'll also include a plus or minus standard deviation associated with that overall mean value. This will allow participants to make direct comparison to NIST values that they measured on the sample materials. Now, I should mention this is a very similar approach that we're planning to do for a cannabis reference material. And the big difference is a cannabis reference material will involve a lot more measurement replicates to evaluate potential biases and sample homogeneity. The initial measurements that have been made at NIST on the CannaQAP hemp oils involve the use of a Cannabis Analyzer from Shimadzu. This is a LC liquid chromatographic separation system connected with a photodiode array detector to allow you to collect 3D graphical data as shown here represented, to where you can have on your x-axis you have your retention time, your y-axis you have your wavelength, and then your third axis is your signal intensity as represented here. And I'm showing you an example of looking at this method focused on looking at 11 cannabinoids. And here I have highlighted here this arrow going over to where you can look at the potential chromatogram and UV spectrum that you can attain from the data collection. Based on the initial measurements and 3D chromatograms that I showed on the previous slide, NIST elected to do a LCEV measurement at a single wavelength. For this, 220 nanometers provided a good, strong signal for all 11 cannabinoids, as you can see in the example chromatogram obtained from a reference standard. In this case, separations obtained majority for the 11 cannabinoids with some overlap for a couple of the analytes. But the resolution is above 1 in all cases and allows for adequate quantitative measurement. Now, the system allows you to also get to calculate a quantitative number for you directly. In this case, you can get a calibration curve, as I have here on the right as an example, looking at 5 calibrants. In this case, you got a calibration range using external calibration with an R-square value of 0.9999, which will lead you to believe you have an excellent linear correspondence over that range. Now, the one thing I tend to caution people when they're making measurements, especially in the case of hemp samples where THC could be at a very low concentration, in your calibration curve, majority of the time, your THC correspondence is going to be at the very low range of your calibration curve. As I have highlighted here, the lowest 3 points in that calibration curve, you'll see that the line actually does not fit very well to these bottom points, even though I have an excellent R-square value. That's because it's being heavily influenced by the higher point in the calibration curve. So careful attendance will be paid on the lower range of the calibration curve. Here's an example of what the calibration curve looks like if you use a narrower range. In this case, I'm using the lower 3 calibration points. I've removed the higher 2. And here I'm getting an R-square value of 0.999 like I did before. except in this case the line is going through all three calibration points evenly and allow for a more accurate determination at this lower range. For the initial evaluation of the LC-UV method, NIST obtained commercial hemp oil samples from 4 different sources ranging in different levels of CBD concentration. For the commercial oils, I'm showing here on the left the procedure used for sample preparation. So if the sample was kept in cold storage prior to analysis, it was allowed to equilibrate at room temperature for 1 hour. 0.5 grams of that sample was weighed out into a 50 milliliter centrifuge tube, and methanol was added to the 50 milliliter mark. The sample was vortexed for 10 seconds, shaken for 15 minutes if necessary. Some of the hemp oils did not require additional shaking as it was immediately mixed between the oil and methanol. At completion of the mixing, the samples are diluted based on a 10-fold and 100-fold dilution using methanol. Here on the right, I'm showing an example of what one of the chromatograms look like. You can see 5 cannabinoids were identified in the chromatogram. Here you have a large peak for CBD, which is typical for a hemp oil sample. And then you have a small peak for delta-9 THC that, if you expand out here on the calibration curve, actually is on the very low end of your calibration curve. I should mention this was a 10-fold dilution measurement shown in this case. The concentration was determined to be 3 milligrams per liter, which is approximately 0.15% delta-9 THC based on the mass of this particular sample. So this kind of highlights the importance of what I was mentioning before about having that lower, narrower calibration range. Now, one reason why it's important for you to include some type of dilution in your sample preparation, even though this makes a very dilute signal, is because the more dilute the sample matrix is, the less likely you are to have matrix interference in your chromatogram, but also potential issues on the instrumental side where you're not putting as dirty of a sample into your instrument. Here's a direct comparison for one of the hemp oil manufacturer samples. In this case, I'm looking at the CBD mass fraction using the label claims on the product versus the NIST LC-UV measurements discussed in the previous slide. For samples 1, 2, and 3, there's a very well comparison between the NIST values and the label claim. Sample 4, the more concentrated sample, the NIST value is slightly higher than the label claim, but it's really hard to make the comparison without some type of statistical uncertainties for the label claim values. On this slide, I'm showing an additional comparison for second hemp oil manufacturer samples for CBD mass fractions. In all 3 samples shown here, the NIST value was significantly higher than the product label claim. The slides up to this point have focused on exercise 1 with hemp oil. But I also want to expand on what we plan for exercise 2, which will include cannabis plant materials. And our initial plan is to start the whole scheduling process as what I showed you before for the hemp oils for a hemp plant for exercise 2 starting in August-September timeframe, where we would hope to be able to ask for participants to start to sign up in October and November for exercise 2. The cannabis samples used for exercise 1 will be multiple samples with concentration that range for THC from 0.1% to 2%, varying to adapt to whatever requirements there could be possibly throughout the U.S. For forensic laboratories to be able to meet their necessary needs. Now, in this case, NIST will have to make sure to provide a homogenized sample, which will be conducted through the use of a specs sample prep cryo-mill grinder to provide an ultra-fine powdered material. Now, this might not necessarily be the exact same type of grinding material sample that would typically be used in forensic labs or cannabis testing labs, but to ensure the homogeneity throughout the entire sample batch, NIST is making sure that that is done through the grinding of the material. And this is a similar approach that we would apply for a cannabis reference material as well. Now, the benefit in exercise 2 of using a plant material is, in this case, we're going to have a more complex sample preparation involving some type of solvent extraction through mixing and sonication and other possibilities. In this case, we're doing an entire method evaluation sample prep along with the different instrumental methods that in the first exercise we were just primarily focusing on the instrumental method. For initial method evaluation at NIST, we reached out to the University of Kentucky Hemp Proficiency Testing Program and obtained some of their hemp plant material samples for initial evaluation. And here I'm showing you an example of a table obtained in the certificate of analysis of the reference sample. These values were assigned using 4 different analytical measurements based on a various of 30+ labs in their study. The sample preparation procedure for the hemp plant material is shown here on the left of the slide. This sample preparation was previously approved by AOAC International and is published in the reference shown below. The same publication was also used for the method for the hemp oils discussed previously. In this case, the samples from University of Kentucky were allowed to equilibrate at room temperature for 1 hour. The samples are normally recommended to be stored at cold temperatures. 0.5 grams of the plant sample was weighed into a centrifuge tube. 20 milliliters of methanol was added, vortex for 10 seconds, and shaking for 30 minutes. After centrifugation of 5 minutes, the extract was removed, and the same procedure was repeated again with 20 milliliters of new methanol extract. The extracts were combined, and then not shown here in the diagram is an additional 10-fold and 100-fold dilution step like we had similar with the hemp oil sample. Here on the right, I'm showing one of the example chromatograms obtained from the Cannabis Analyzer, showing we were able to identify 9 cannabinoids in the sample. In this case, here I'm focusing on the delta-9 THCA peak, where we had a concentration of roughly 3.5 milligrams per liter, which was associated to roughly 0.3% delta-9 THC. So this material allows us to evaluate the method using a sample right at the federal limit. On this slide, I'm showing a comparison between the University of Kentucky PT participants' results and the NIST results for the LC-PDA method. Both these samples were from the September PT study at University of Kentucky. In this case here, there's generally a good comparison between the NIST value and the participants' values associated here within the uncertainties provided for the analytical measurements. Here's an additional comparison for a second set of samples obtained from the University of Kentucky, where NIST values and the PT participant values represented a good overall agreement for the different analytes targeted in this study. The extraction procedure used so far in these studies have been shown to provide accurate results. However, it has a major disadvantage of sample extraction procedure currently lasting 70 to 80 minutes. Now, the advantage of the procedure is that you can do multiple samples at one time to kind of offset that. But in a forensic laboratory where they're performing measurements on a very large number of samples, a 70-80 minute extraction procedure would not be appropriate for them. So one of our goals is to try and optimize the extraction procedure that we've already started to use to target a 30 minutes or less time frame through the use of evaluating extracting solvents, solvent volumes, and extraction times. In addition to the LC-UV methods discussed previously, NIST will be looking to use LC tandem aspect for assigning values to CannaQAP materials and cannabis reference materials for multiple different cannabinoids. Here I'm showing you an example of an LC tandem aspect chromatogram that uses a multiple reaction monitoring mode. In this case here, when you start to incorporate mass spec, it'll be important that the user include a isotopically labeled internal standard. In the case of delta-9 THC, you would want to use the isotopically labeled D3 component or a D9 component that is available on the marketplace. The use of the MRM transitions allows for a simpler chromatographic representation of the sample. It helps to eliminate a lot of the matrix effects that you can commonly see in using single ion monitoring for LC measurements. NIST will also develop a GCMS method for assigning values on CannaQAP materials and reference materials for cannabinoids. Typically in GC, a derivatization step is implemented to allow for the analysis of the acid form and the neutral form of the cannabinoids. This is important because during the sample inlet on the GC, which is usually done at temperatures of 225 degrees C or 250 degrees C, the acid form will automatically be converted into the neutral form in that process, preventing the ability to measure both analytes in the chromatogram. So here on the left, I'm showing you an example when you have the derivatization step implemented, where in this case you're going to get a delta-9 THC peak separate from the THCA peak. But when you don't use the derivatization step, you get one peak in your chromatogram that represents the combination of both analytes. Now, part of the issue in this case is that you don't have 100% conversion in the GC inlet, which can cause an issue when you're trying to make a quantitative measurement. The GCMS instrument is typically operated in either full scan mode or SIM mode. In the case of full scan mode, it's very important that you make sure to get a baseline separation of delta-9 THC and its stereoisomer delta-8 THC. As you can see here on the right, the full scan mass spec of the two isomers are very similar to each other. Now, the main difference in the two mass specs are the mass charged ions at 299. In the case of the delta-9 THC mass spec, it is the highest abundance peak. In the case of the delta-8, it's a fairly small signal representation. So in the case of SIM, if you use this ion mass charge 299, you're going to eliminate a lot of the presence that you might see from delta-8 THC, and that would be included in the method that's developed at NIST. I would like to thank you for your attendance today, and I hope you learned a lot about what we're starting to do at NIST, developing tools for the cannabis community, and what we're also doing on the method development side currently and in the future. And I'd like to acknowledge some of my colleagues in the MML laboratories at NIST that are part of the cannabis team, the group that I'm part of, the organic chemical metrology, who are going to be doing the cannabinoid measurements, but also we're going to be, I haven't mentioned, but we're going to be focusing on pesticides, mycotoxins, a wide variety of different analytes. The inorganic group is also part of the team, where they'll be looking at making toxic metal measurements as well in the future. And then we also have another group on the forensic side, the surface and trace chemical analysis group, that are developing a colorimetric test for looking at variation between THC and CBD. I would also like to acknowledge our partners for CannaQAP Exercise 1 sample development at CV Sciences and Alchemist Labs for providing initial measurement evaluations of the materials. For additional information on CannaQAP or cannabis reference materials, you can email the cannabis team at cannabis@nist.gov. For particular information on the participation in CannaQAP, here's the website available for you to click on. And then we also have a general cannabis research page that we're in development of now, and the link is shown here on the slide. And with that, again, I'd like to thank you for your attendance today, and I look forward to answering some of your questions in the Q&A portion, getting ready to start. And with that, I would like to turn it over to Rachel.
Let's thank both Melissa and Brent for such exciting presentations. Again, shown here are the helpful websites if you want to learn more information about NIST programs or Shimadzu's analytical solutions for distinguishing between hemp and marijuana. If you have not had a chance, please fill out the survey questions on the left-hand side to provide feedback and request additional information. At this time, we will begin our Q&A session. Thank you to the audience for attending and sending questions. Looks like we have some time to answer a few questions.
All right. Thank you all for attending. We have a few questions that we are going to get to. First question, do you have matrix blanks available for potency or terpene testing?
Thank you, Rachel, for a good question. Currently, we do not sell a reference material for that direct purpose. I know that in the cannabis industry, this is something people like to use for when they spike stuff in for analysis, but currently we do not have a no THC or terpene reference material.
Thank you. Brent, another question for you. Why did you select the Shimadzu LC for this analysis?
The Shimadzu instrument was selected because it's being advertised out to the cannabis community and forensic community as an all-in-one package where it comes with predetermined methods, reference standards, so everything comes in one package. Although it is an LC-UV PDA system, other manufacturer instruments could work. Ideally, the method could be transferable between multiple instrument manufacturers.
Rachel, do you think you could tell us a little more about what's included in the cannabis and hemp analyzers and what the difference is between the two?
Yeah, absolutely. So the cannabis or hemp analyzer is actually the same thing. One is just marketed towards the cannabis growing industry and the other versus the hemp growing industry, as they are two separate based industries. Each of them will come with your standard methods. We have 3 different method packages that are available. These are high resolution, high sensitivity, and high throughput. In addition, all of your standards, a 10, 11, or I believe we now have a 16 mix are available, as well as your mobile phases and any columns and pre-columns that would be needed are all included in the analyzer as one package. So it can be placed on the bench and run, you know, as simply as the next day after checkout specs. Thank you for that. Another question. Will the full method for sample preparation with sonication be made available?
So the method is actually currently available. The method I used was taken directly from the publication listed there on that slide. In 2018, an expert review panel at AOAC International approved a method for high THC marijuana samples. The only difference between my method that I use in the slides and what's published is I use methanol instead of ethanol. So I would recommend to refer to that manuscript. I would also, since I mentioned AOAC, I would like to mention to the attendees that there are multiple working groups within AOAC, one focused on cannabinoids, one on proficiency testing, contaminants, and I would recommend to everybody, if they're wanting to find out more information or be more involved, to reach out to try and join one of those working groups.
Thank you, Brent. Another question, I think this ties into the methods that you guys are looking into. Will the LCMS method be single or tandem? And what GCMS and LCMS instruments are you currently using?
So yes, the method we're going to be developing for LC mass spec will be a tandem mass spec method. The idea is that the method would be transferable between an Agilent system, a Shimadzu system, or Thermo. So multiple vendors, multiple instrument platforms should be able to use the same method. In the case of GC, it's the same thing as trying to develop methods that would work. The ones that we're using now is currently an Agilent instrumentation.
Thank you. So for labs interested in participating in CannaQAP studies, what time commitment should be expected?
So typically we give the participants around 6 weeks to make measurements on their part. So I would say once you receive the samples, there will be enough of the material for you to do some practice measurements with before you actually make the measurements that you want to submit. But I would say no more than probably a week's worth, if that much. It really depends on how much the work you've done beforehand on developing your method.
Thank you. And what is the percentage of THC to consider it drug type not hemp?
So according to the federal regulations, that if it has a percentage of 0.3 or greater than 0.3%, it's considered marijuana, not hemp. Now that is the federal regulation. Some of the states have different regulation on that, and it goes from state to state. I've heard that some are 1%, so it could vary depending on where you're located. And also, if you're outside the U.S., I know there's definitely differences in the concentration threshold.
Okay, thank you. This is an interesting question about another material. So I find that oils all have varying solubilities and give different extraction efficiencies. How do you account for this with internal standards?
So if you have a internal standard and you spike it in at the same time that you're prepping your samples, so basically the internal standard goes through the entire process of if it doesn't solubilize right away, and you do some type of sonication or shaking, it's going through the whole process similar to the THC or cannabinoids in the sample. Now, I would point out the best internal standard used would be an isotopically labeled. I know that there are methods out there that don't use isotopically labeled. So the main thing is just make sure that you get good representation with whichever internal standard you choose.
Great. Next question. What is the criteria for peak ID in the methods?
Okay, so for the LCUV method you saw was based off of two identifications. One was the retention time comparison to an authentic reference standard, and the other was the UV spectra that corresponded with that cannabinoid. Now, in the case of GCMS, if you're doing a full scan mode, the identification is done based on the retention time again. but also you can do a full scan mass spec search using the NIST database, and that allows you to confirm that the peak, let's say you're looking for THC, is THC. Now, in cases where you're using a sim mode, you have to vary some on what you're doing for identification. The retention time will still be the same, but in the sim mode, you currently, if you're just using one ion, you're just going to see the one peak in your mass spec. And then for LC tandem mass spec, you're going to have the retention time again, but you're also going to have the transitions from the product ion to the precursor ion that allows you to identify THC because the chances of there being another cannabinoid or analyte in that sample matrix that mimics that is going to be on a very limited basis.
Thank you. Did you evaluate the effect of derivatization on sensitivity or selectivity?
We have not evaluated that as of yet. My answer to that question really deals with if you're using an internal standard, then keep in mind that your internal standard, if you're using isotopic labeled and it's tagged with the same derivative, you should account for any variations that you might see in the process. But to the actual signal strength that you might get, or we have not addressed that at this time.
Okay, thank you. Next question here. What methods is NIST using for identification of untargeted substances of abuse in cannabis, such as synthetic derivatives?
Good question. At this time, we haven't done any non-targeted analysis for substance of abuse in cannabis dealing with synthetics. That is something that we would like to do, but right now the focus has been on trying to get a reference material and a CannaQAP started up using plant materials where we're not really focusing on a non-targeted approach at this time, but we do have interest.
Okay, will you be working with the Cannabis Commission on the determination of specification limits of cannabinoids and products?
So we, all of our data and methods that we do will be public, and a lot of the data that we get from CannaQAP and that goes into the reference material will all be made available public for anyone to have access to. Currently, right now, we're not working with Cannabis Commission, but I'm definitely open to having a conversation with them and talking about some type of collaboration.
Rachel, this next question's for you. Can the marijuana analyzer that be used to test for hemp, or do you have to have the specific analyzer for specific product types?
That's a good question, Melissa. It is, as Brent stated in the presentation, an HPLC, PDA, or UV detector. And so the marketing is that it is for cannabis or hemp, but either could be run on the system. So it does not have to be the cannabis has to run exactly cannabis. You can run hemp on it. Great question. This question's for Brent. For the sample prep, what is the difference between vortex and shake?
So vortex is a very high speed that I did just for 10 seconds to remove the powder from the bottom of the centrifuge tube, and then I put it into the shaker. Most likely, you will just shaking it over time, you're loosening all of the powder that's stuck to the bottom of the tube. It was just a way of making sure that it's done right away.
Okay. And Brent, why did you select the Specs grinder / mill? Are there any other grinders that are acceptable to use for the sample preparation?
That is an excellent question. So the reason why we chose the specific powder mill grinder which I think is primarily the only one that's available is through specs, is because it's the cryomill to get an ultra-fine powder. It's a little different from necessarily when you're in a forensic lab or a cannabis testing lab. You might not necessarily need as a ultra-fine powder, but when you're trying to make a reference material or a cannabis sample that's homogeneous throughout the entire batch, because keep in mind, a reference material we'd probably be looking at trying to develop, like I would say 50 pounds or more. So we need to make sure that the homogeneity throughout that entire batch would be accurate. So that's the reason why I chose that particular cryomill grinder. I do know that some labs out there have been or looking into using coffee grinders. And I should also mention that Specs does sell a couple other grinders that aren't cryomill that do a really good job of grinding the material as well.
Thanks, Brent. We have a question for Rachel about distribution of these instruments. Can the Cannabis Analyzer and Hemp Analyzer be shipped to the Middle East?
That is a great question. Thanks, Melissa. So we have many subsidiaries all throughout the world for Shimadzu Corporation that can assist you with these particular systems. So if you're interested, reach out, fill out the survey information, and we can provide more information for you about that to the Middle Eastern countries. This question is for Brent. How do you sign up to participate in the CannaQAP program?
So there should be a link on that last one of the last two slides that actually will take you to the CannaQAP website, and there's a way there you can just request a new account, and that will send an email to us notifying that a new account has been created, and we can go in and approve it. If for some reason you weren't able to get that link, you can also just email us directly at cannabis@nist.gov, and I will more than happy to forward you the link to the website so you can go in and create an account.
Great. Here's a couple of questions. These kind of tie together. So where did the value of 0.3% THC come from? Do you know the reason? And then regarding that rule, what are you using for an uncertainty value?
So to answer the question about where did that number come from, to be honest, I don't really know. That was just what was stipulated in the Farm Bill. I do know that, again, that number varies, like if you go into outside the U.S., but I don't know the detailed backstory to why that particular number was chosen. Regarding the uncertainty value, so when we typically do a reference material, we make usually many replicate measurements using multiple instrumentation, and we don't use the standard deviation per se as what the uncertainty is. We have statisticians that will go through and enter it into certain statistical programs to allow us to be able to determine an expanded uncertainty that will be associated with that. But that takes into account the analytical method is being used, the sample prep, the number of replicates, and everything gets taken into account on determining that uncertainty value. But normally we do provide you with a way of determining what the standard deviation is from that expanded uncertainty.
Great. All right. So it looks like we have time for one last question. So what is the ratio of THC to CBD, CBN to distinguish between hemp and marijuana?
Really, there is no ratio to per se that will definitely tell you if it's marijuana or hemp. I know that there are some colorimetric tests out there that are based on a 1% on a ratio of 1 of hemp CBD to THC. Really, the only way to know if it's hemp or marijuana, you need to quantitatively measure THC in the material and see if it's below or above the 0.3% threshold. And also, I would like to mention, since that was the last question, I'd like to thank the attendees for being here today. I hope you learned some information from the presentation, and please feel free to reach out to us if you have any questions, or I'm sure Rachel will be more than open to. And with that, just again, thank you for being here today.
Thank you, Brent. Looks like we're at the end of our allotted time for this Shimadzu sponsored ASMS webinar. As Brent had said, we'd like to thank you, the audience, for participating in all of these very interesting questions. If we're not able to get to your question today, we'll reach out via email to answer any additional ones. And once again, I do want to thank our speakers, Brent Wilson and Melissa Phillips, for these presentations, and for you for attending. We will send out an email with a link to view the recorded version of this presentation in the next day or so be on the lookout for that. We hope to have you back in webinars that we organize in the future, and have a great rest of your day. Thank you.